Good morning, students. So today we are going to discuss about the overview of pathogenic microbiology, right? I hope that everyone is able to see the screen. So now I'm going to start the class that is on the overview of pathogenic microbiology. So first of all, I would like to tell you that what is pathogenic microbiology. So basically, pathogenic microbiology is a branch. As you know that microbiology is a branch in which we deals about the study of microbes like virus, parasites, bacteria, fungus, algae. So when we talk about the pathogenic microbiology, it's about those microorganisms which are harmful to the humans, right? Basically they have the pathogenic capacity. It means they are able to cause the infections to the human. So the study of those microorganism which causes the infections, which causes the severe diseases to the humans comes under the pathogenic microbiology. Because see, microbes are two types. They are the pathogens or they are the commensals. So commensals are those microorganisms which are the normal flora of our body, means they are normally living in our body, and in return they don't cause any harmful effects on our body. They provide benefits to us. And other classes, pathogens. Pathogens are those microorganisms which cause disease to us, and also they are using our nutrition, they are living inside our body, and in return they are causing infections to us. So here is two classes, pathogens and commensals. So when we are talking about the pathogens, it comes under pathogenic microbiology. It means they are those microorganisms which cause disease to the specific person or which cause specific infections to the humans, right? So let's start with the pathogenic microbiology. See, it is the study of microbes that cause diseases. It focuses on how they infect the body, how the body responds against the infection, how we are able to diagnose the infections, and how we are able to treat or prevent the infections. So in this subject, we are going to learn about the study of microbes that are pathogenic. Only pathogenic microorganisms you will go to learn in this class. And what about the pathogenic microorganisms? It's basically about the mode of infection, that how they spread the infection, how our body responds against that particular infection, and what are the techniques, what are the lab diagnosis, and what are the laboratory diagnostic technologies we have with the help of we are going to diagnose them, and how we are going to prevent them, what are the preventive measures we have to take to prevent the spread of that particular infection. So why we need the pathogenic microbiology? So students, to understand the need of microbiology, we have to go to some points like, as the patient is infected with a causative agent, and the patient is carrying that particular causative agent. So to identify the root, to identify the cause of the disease, we have to done the laboratory tests like your microscopy, your culture, your staining. So to identify the disease causing microorganisms, we need pathogenic microbiology. To understand how infections occur and spread, that what are the routes of transmission of infections, and in after coming in our body, what route the commensal, what route the pathogen takes to infect our cells. So to understand that how infection spread and how infection progress, we need pathogenic microbiology. To develop treatments and vaccines, we need microbiology. To give the preventive and control against the infectious disease, we need pathogenic microbiology. And also to improve the health of the public and for the safety concerns of the population, we need pathogenic microbiology. So basically it is the branch of science which deals with the safety, prevention and treatment of the humans. So in the Unit 1, you will going to read about the infectious diseases. That what are the infectious diseases, how they are spread, how they are recognized, what are the symptoms from which we are able to recognize the infectious diseases, and what are the different types of the infections caused by the microbes, what is the disease cycle, how the disease progress in the particular human beings. So in the syllabus, first is the terminology. So in terminology part, you will have to learn about the infections, that what are the infections, what are the pathogens, what is the host, and what is the carrier. So in simple words, we can say that infection is the process which develops in the body when the pathogen comes in your body, right? When you infect with particular pathogen, so the pathogen produces some harmful effects on our cell, then it is termed as infection. And the causative agent which is responsible for spreading the infections, which are responsible for producing the harmful effects in our body, is the pathogen. Next is the host. Host is the individual who harbors the pathogens, right? Host, who are host? Host is the individual who harbors the pathogen, who provides the shelter, nutrition to the host, to the pathogen for their survival. And one is the carrier. Carrier is the individual who carries the microorganism in the body without any symptoms. Like, see, we have two patients. One is the host and other one is the carrier. One is the infective person and other is the carrier. So the infective person is having the symptoms of the diseases, but carrier doesn't have any kind of the symptoms, but it carries the pathogen in the dormant state, right? Next is the frequency of disease, that how frequent the disease spreads. So the pattern of the spreading of diseases, like it, first of all, it is sporadic, then it converts into endemic, then epidemic, and then it's pandemic. Sporadic means it is limited to one person. Endemic, it means that it is limited to a particular area. Epidemic means it is limited to the whole country, and pandemic means it is not limited to any country, not limited to any area. It is spread all over the world. Then it is termed as pandemic. Next is about the recognition, then that how you are able to recognize the diseases. So to recognize the disease, we have to identify the signs and symptoms of the patients. For example, if anyone is suffering from malaria, so he or she is experiencing high fever, frequent fever, fever with severe chills, weakness, lethargy. So, by understanding the signs and symptoms of that particular disease, we are able to uh, uh, prognose this condition of the patient. On the basis of prognosis, we are able to we can say that we are able to identify some laboratory tests. We are able to request some laboratory tests so that patients will confirm about their clinical condition. So we have to prescribe some lab tests to the patients to identify that in actual the patient is suffering from which kind of the infection, and the patient is carrying which microorganism, right? Next is the infections and diseases cycle. So in this part, you will learn about that how the infections spread in the human bodies. So in short, if I'm telling about the infections, if I'm told you about the infections, so it if I'm tell about the infections, so first the bacteria, the virus starts attachment with the host cell. First of all, they attach with the host cell, and then after attachment, they insert their genetic material, they multiply in our body, and they undergoes in proper incubation as per their need, and in the incubation period, they multiply and they increase their size and also increase their number. After increasing their numbers and the size, they produce specific illness and they produce toxins which cause the which causes the host cells to lyse and spread the infections, right? After spreading the infections, once the host nutrition is diminished or deplenished, their count decreases and they start dying. This is known as decline phase. Next topic which you have to learn, that is virulence. Virulence is the power of the any microorganism which defines the disease causing capacity of the microorganism, or you can say that it is the degree of pathogenicity, that how much pathogenic a microbe is, right? This is known as virulence. Next is mode of transmission, that what are the routes which a microbe choose to transmit or to go into the body of the host. So the infections are spread from direct contact. For example, a healthy person comes in the contact of come in contact of an unhealthy person. So from the unhealthy person, the healthy person acquires the infection. This is known as direct contact. When two persons come in contact, one is healthy and another is unhealthy. So from unhealthy person, the healthy person acquires the infection. This is known as direct contact. Second is by the air. So if we are acquiring infection from air droplets, this is known as air route. Next is water and food. So if you are getting ill by ingesting the contaminated food and water, this is known as fecal-oral route. It means the food, the water you have intake, it is contaminated with the microorganism, the pathogenic microorganism. So with the ingestion of food and with the ingestion of water, the microbe content will goes in our body and it goes to your body, and the bacteria start and the virus start multiplying and the person is ill. Next is the vector. So here we have some vector-borne diseases also, like malaria, your filariasis. So in the malaria and in the filariasis and the dengue, the vector, the mosquito is responsible for the transmission of the disease. So the diseases which are spread or which are transmitted by the bite of the vectors, they are comes under vector-borne diseases. Next is emerging and re-emerging disease. Emerging and re-emerging disease means emerging infections are those infections which appear in the body of the patient on first time, and re-emerging infections are continuously returning in the body of the patient. So they are the re-emerging infection. For example, if a person is having any kind of the infection and the person is cured after some time, but after time, after the passage of the years, he or she again develop the same infection. They are known as the re-emerging diseases, like COVID-19, tuberculosis. Next is that which countries are the major source of spreading the infections, because some of the microorganisms are originated from different countries, right? So they have the higher chances of the cases and higher chances of having that particular diseases, which comes under global travel and health. Next is the nosocomial infections. Nosocomial infections are those infections which are acquired from the hospitals. That's why they are also known as hospital-acquired infections. For example, if a patient is undergone a surgery, he is having the stone in the gallbladder, and the doctor and the OT technician plans the gallbladder surgery, like cholecystectomy of that particular patient. Patients undergo the surgery, he is not having any kind of the infection, but during the surgery, the instruments which are used over the patient, they are contaminated, and the patient got the microbes injected in the body. So when he goes to the home after the surgery, after the incubation period, he developed the infection. So the nosocomial infections are those infections which are acquired from the hospital, which are not present in the person before admitting to the hospital, right? So the infections which appears in the patient after admitting in the patient, they are the nosocomial infections. So let's move toward the unit 2. So students, let's summarize the unit 1. So as I told you that in the unit 1 you have to learn about the virus, microorganism, what are the different kind of the microorganism, what are the different kind of pathogenic microorganism, how they transmit from one body to another, how they multiply in the body, what are the routes of their transmission, what are the emerging infections and re-emerging infections, and also what are the nosocomial infections and what are the causative agents of the nosocomial infection. So these are the topics that you have to cover in the unit 1. Next is the unit 2, that microbes of medical importance. So some of the microbes are pathogenic, but see, as well as the pathogenic, some of the microorganisms are also helpful to us because they are help us in the medical lab. So how they are helping us and why we need them and why we need the normal flora, let's see. So in this unit you will learn about the nomenclature, that how the name of the microorganisms are discovered, and so the name of the microorganism have two things. First one is genes and other is the species. For example, the causative agent of the of the tuberculosis is Mycobacterium tuberculosis. So the Mycobacterium is the genes and tuberculosis is the species, right? Genes have multiple kind of the species which are responsible for different kind of the infections. Next you will learn about the classification. In the classification you will learn about the different types of bacteria, different types of the viruses, different types of the fungus, different types of the parasites, based on their structure, based on their cell shape, based on their cell size, based on their mode of nutrition, based on their mode of infection, based on their routes of transmission, based on their genetic material, based on their pathogenicity, right? So on the basis of the structure, shape, cell size, cell type, functions, genetics, and mode of nutrition, we have multiple type of the microorganism. Next is the normal flora. Normal flora are those microorganisms which are present in our body from birth, or we are acquired from the environment, food, and the contact. Let's see some of the examples of the normal flora. So see, Staphylococcus and the Corynebacterium, both are the pathogenic bacteria, but they are the normal flora of our skin. Staphylococci epidermidis is the normal flora of our eyes. Streptococcus and Neisseria is the normal flora of our respiratory tract. Streptococcus and Lactobacillus is the normal flora of mouth, and also Lactobacillus is the normal flora of vagina. E. coli and Bacteroides is the normal flora of intestinal tract. Lactobacillus and Staphylococcus is also the your normal flora of urinary tract, right? And unit 3 is about the mode of microbial infections, that how the microbial infections appears in the body. So first one is microbial adherence. See, if a microbe wants to infect your cell, we have several steps which a microbe has to fulfill. First one is adherence, second one is penetration, next is active penetration, then after penetration it will goes under multiplication. After multiplication, the microbe spreads to the other organs and it's challenge to your immune system, and also starts the damaging of your tissues. And once the damaging of your tissues starts, you feel some symptoms. So you guys are not able to see the change slides? अभी हो रही है स्लाइड्स चेंज क्या? One more thing, those students creates nonsense in the chat box, I will remove them. Just wait for 5 minutes. And they are not allowed to join again. डबल 549 133 ये सीखते हो आप क्लासेस में? 5549133 आपका कम्युनिकेशन का ये मोड है क्या? कल को हॉस्पिटल में जाके पेशेंट्स के साथ ऐसे बात करोगे? If you are not able to respect your teachers, you will never succeed in your life. अभी अगर मैं माइक अलाउ करूं और कैमरा अलाउ करूं, एक वर्ड नहीं बोल पाओगे तुम. What sorry? बकवास करने के लिए क्लास ज्वाइन करते हो तो मत करा करो. कोई जरूरत नहीं है क्लास में आने की. सीखोगे भी क्या क्लास से? खाना ही बनाना है तो घर पे बना लो. क्लास में क्यों आना है? You are not meant to be a paramedical. आप लोग बने ही नहीं हो पैरामेडिक कोर्स करने के लिए. Just leave the class. 5549133 लीव कर दो क्लास. और जाके खाना बना लो. Get out from the class. Are you guys are now able to see the change slides? Okay. See, I will not tolerate any kind of nonsense in the chat box. If you want that chat box is open, then please don't write anything in the chat box. Otherwise, we will not provide any classes to you. See, in the introduction, we have learned that microbiology is this branch in which we learned about the microbes, basically about the bacteria, virus, fungus, parasites, right? So in the pathogenic microbiology, we deals that how the microbes infects the patients, infects the host body, and they are spreading the infections from one part to another, and what are the modes of transmission, how the pathogens comes inside the host and spread the infections. So the need of the pathogenic microbiology is that they are responsible to identify the disease causing microorganism. They understand that how the infections occurs and spread, to develop the treatments in the vaccines and to prevent and control the infectious diseases. So in unit 1 we have seen that we have some terminologies, we have some virulence factors, we have some modes of transmissions, we have emerging and re-emerging infections, and in unit 2 we have seen that the to nomenclature, to provide name to any particular bacteria, we have the binomial system in which the genes and species are collectively combined. For example, if the causative agent of tuberculosis is Mycobacterium tuberculosis, so the genus is the Mycobacterium and the species is tuberculosis. The genes have multiple kinds of the species which is responsible for producing different kinds of the infections, right? So next is the classification. In the classification, they classify the microbes on the basis of their shape, on the basis of their cell type, on the basis of their cell structure, on the basis of their mode of nutrition, on the basis of their genetics, and on the basis of their pathogenicity, right? Next is the normal flora. So normal flora are those microorganisms which are present in the body of a human being from birth, right? They are present inside the body of a normal person from the birth. For example, in the skin, we have Staphylococcus and Corynebacterium diphtheriae. In the eyes, the normal flora is the Staphylococci epidermidis. In the respiratory tract, the normal flora is Streptococcus and the Neisseria. In the mouth, the normal flora is Streptococcus and the Lactobacillus. In the intestinal, the normal flora is the intestinal tract, the normal flora is the E. coli and bacteriotes. And in the urinary tract, the normal flora is Lactobacillus and Staphylococcus. So these are the normal flora which are present inside the body in normal conditions. See, now the class in the English only. If you want explanation in the Hindi, just wait. Once I will over the class, I'm come to take your doubts. In the doubt session, I will explain you in Hindi also. Is that clear to everyone? Just collect your doubts after explaining the slides. I'm come to your doubts. Next is the mode of microbial infections. See, mode of microbial infections. So that how a microbe is able to cause the disease, that how it transmits into the body and how the microbe is responsible for causing the infection. So to cause the infection, the first step is adherence, right? On number one, a microbe is attached to the host cell. To cause the infection, the first step is adherence. So in adherence, a microorganism adhere to the host via special organs like pili, fimbriae, adhesion proteins, right? On the cell membrane of the microorganism, for example, in the bacteria. In the cell membrane of bacteria have some special structures like pili or fimbriae. So fimbriae or pili is used for the attachment with the host cell. Fimbriae or pili is like a bridge between the microbial cell and the host cell. So by means of fimbriae or pili, a microorganism is able to attach to the surface of host cell. This is the adherence. Next is passive penetration. First one is direct adherence. Second one is passive penetration. Like microbes enter the host body through any open wounds, through cuts, through breaks in the skin, or through mucous membrane. For example, if you have cut in your finger, so there is an open wound. So from the open cut, from the open wound, the microorganisms come inside the body. Next is active penetration. In the active penetration, after entering in the human body, the microbe starts to produce enzymes, right? Once they comes inside the body, they starts to produce the enzymes, and enzymes are responsible to infect the tissues. It's the enzymes like hyaluronidase, collagenase. So these are the specific enzymes which are produced by the microbes which infects the tissues, and they starts the multiplication. After penetration, they starts multiplying, and as their numbers increases, they spread from one part to other part, and the spreading of the infection continues. Once your immune system sees that any other antigen comes in the body, the immune system also activates and produces some antibodies to fight against the particular infection, and till time the microbe continues to damage the cells and ultimately damage the tissue, and the patient experience symptoms, right? And what are the factors does a microbe have to infect the tissue or to spread the infection? That is having the virulence factors. And what are the virulence factors? The virulence factors are the toxins, the enzymes, the capsules, and the mechanism which the microbe have to destroy the immune defense of the host. Host is the individual who carries the microorganism, right? It is the individual who harbors the pathogenic microorganism. Is the person who provides the shelter, nutrition to the microorganism. Microorganism use the nutrition and take the shelter from the host and multiply in the number and produces its toxins, enzymes, capsules, and start affecting the other cells or tissues. See, if I'm talking about the flow chart, so see that how the microbial infection spread. First one is microbial adherence. First of all, microbe attached to the host cell via special organs which is present over the cell membrane of the microorganism. Either it will comes inside the host body through the open wounds or cuts. First, the microbe enter in our body through open wounds, cuts, through fecal oral route, through air droplet route, and after entering in the body, it starts with the adherence with the host cell, our cells, right? Once the adherence is complete, they start penetration. After penetration, they start multiplication. After multiplication, they start spreading. After spreading, they evade your immune response and start damaging the tissues, right? Once the tissue damage starts, the patients experience signs and symptoms, and the factors which are responsible to produce the symptoms, they are the toxins, they are the enzymes the bacteria have, they are the capsules the bacteria have, they are the mechanisms the bacteria have to break the host immune system, right? The next unit is about antimicrobial drugs. See, antimicrobial drugs are those chemical agents which are effective against microbes, right? Anti means against, microbes means microorganisms. So they are the chemicals which are against the microbes. Antimicrobial drugs are those chemicals which are used to treat the disease, right? So they are against the microbes. Like if I'm talking about bacteria, so against bacteria we have antibiotics. If I'm talking about viruses, against viruses we have antivirals. If I'm talking about fungus, against fungus we have antifungals. And if I'm talking about parasites, we have anti-parasitic drugs. So collectively, we are called them as antimicrobials. Means it is a kind of chemical which is used to treat the diseases, and they are basically against the microbes. Whether it is bacteria, it is virus, it is parasite, or it is fungus. So the chemotherapy, it means treatment of the patient with the chemicals, is given by a scientist who is known as Alexander Fleming. Alexander Fleming is the one who discovered the penicillin. He is the one who gives the world a first antibiotic, that is penicillin. Penicillin is the first antibiotic which is effective against the bacterial infections. And the Alexander Fleming discovered the era of antibiotics. He do many research in the field of chemotherapy and developed many more antibiotics, and from there the chemotherapy era got started, and the researchers and the scientists starts working towards the development of antimicrobials, right? So what are the general characteristics of antimicrobial drugs? See, there are some general characteristics of an antimicrobial drug, like they have the selective toxicity. It means they are only harmful to the microbes, not to the host. It means, see, if a person is infected with malaria, right, and we are giving them the anti-parasitical drug, because malaria is a parasitic infection, so the drug is effective against the parasite only. It doesn't harm the person who harbors the parasite, right? So the antibiotics having the antimicrobials having the selective toxicity. It means they are harmful to microbe only, not harmful to the host. They have the spectrum of activity. It means some of the antibiotics having the spectrum to affect many kind of the microorganism, but some of the antibiotics have narrow spectrum. It means they are used to treat the infections of specific microorganism only. It means, let's suppose one antibiotic is used to treat multiple infections, so the antibiotic have a broad spectrum. It means it is used to treat multiple kinds of the infection. It is effective against many microbes. But if I'm talking about narrow spectrum, it means the antibiotics is effective against only specific microorganism. It is a specific that this antibiotic is specific to that bacteria, that virus only. Clear? So it is about the spectrum. Next is cidal versus static. We have two terms: bactericidal antimicrobial drugs and bacteriostatic drugs. Cidal means, it means they contains the chemicals which kills the growth of bacteria. Bactericidal means they are the chemicals which kills the bacteria. But bacteriostatic means bacteriostatic agent are not able to kill the bacteria, but they prevent or they inhibit the further growth of bacteria. For example, if a person is infected with Mycobacterium tuberculosis and the person is having the TB, if you are giving them the bactericidal drug, it means the Mycobacterium tuberculosis will kill by the bactericidal agent. But if you prescribe the bacteriostatic drug to that patient, it means bacteria is not killed, but it will not further grow. It inhibits the growth only, right? So we have two terms, static or cidal. Static means it stops the growth, and cidal means they kills the growth. Okay. Antimicrobial drugs have minimum side effects to the host, but they are not lethal. They have just minimum side effects like diarrhea, vomiting, and loss of appetite, but they are not lethal. They are not harmful. They are not lethal to the host. Stability, it should remain active in the body and reach the infective site. microbial drugs have the ability, they remain active in the body and they reach to the infectious site and support the immune system and help the immune system to kill the microorganisms. Next, you will learn about the determination level of antimicrobial activity. First one is minimum inhibitory concentration. Minimum inhibitory concentration is the concentration which is required to a bacteria, which is required to a microorganism to inhibit its growth, right? The minimum concentration which you are provide to the host to inhibit the growth of the bacteria. Next is minimum bactericidal concentration. It is the lowest concentration that kills the microorganism. Lowest concentration of antimicrobial drugs which kill the microorganism. Kirby-Bayer disc diffusion method. You will also going to learn about the Kirby-Bayer disc diffusion method. It is also known as antibiotic susceptibility test, to identify that which antibiotic is effective against which kind of the bacteria. We have to run the test which is known as antibiotic susceptibility test. So Kirby-Bayer is one of the method by which we are able to apply the antibiotic dust over the culture plate and we are able to identify that which antibiotic is susceptible to which kind of the bacteria, right? In this method, a culture plate is used on which the growth of the microorganisms are spread in a uniformly motion. Over the growth, we have to apply the antibiotic dust. 5 to 6 antibiotic discs are applied on a single plate, and after applying the antibiotic, the culture plate is incubated in the incubator for about 24 hours. After 24 hours, we will take the culture plate, and after opening the culture plate, you are able to see the zone of inhibition. The antibiotic disc will clear the growth of the microorganisms. So the zone of inhibition is the zone which is used to measure the killing capacity of the microbes, right? E-test stripped with a gradient of antibiotic is also used to identify the minimum inhibitory concentration of the antibiotic. Next is how the microbial agents act, that how a microbial agent is able to treat the or cure the infection. So first of all, antimicrobial agents inhibit the cell wall synthesis of bacteria. They inhibit the protein synthesis. They destruct the cell membrane of the bacteria. They inhibit the nucleic acid synthesis. They inhibit the metabolic pathways which a microorganism is used. So by inhibiting the cell wall synthesis, by inhibiting the protein synthesis, by inhibiting the cell membrane synthesis, by inhibiting the nucleic acid synthesis, we are able to kill the particular antimicrobe, right? So to inhibit the cell wall synthesis, we have penicillin, cephalosporin. To inhibit protein synthesis, we have tetracycline, erythromycin. To inhibit the cell membrane synthesis, we have polymyxin. To inhibit the nucleic acid synthesis, we have quinolones, rifampicin. To inhibit metabolic pathways, we have sulfonamides, which block the folic acid synthesis. And also you have unit 4 in your syllabus, which consists of antimicrobial drugs. That what are antimicrobial drugs? And as I told you that antimicrobial drugs are those drugs which are the chemicals which are effective against the growth of microorganism, or we can say that which kills the growth of microorganism. So next is the factors that influence the effectiveness of antimicrobial drugs, that how antimicrobial drug is too much effective. So what are the factors that influence the effectiveness of antimicrobial drugs? First one is the drug concentration, that how much drug concentration you are provide to the infected person. Route of administration, which routes you have used to administer the drug. Presence of pus of necrotic tissue, patient immune status, microbial load, bioavailability and metabolism of drug, microbial resistance, right? So if you follow these factors, like if you have an idea that what is the minimum drug concentration which you have to give to the patient, it will also effective. If you know the correct route of administration of the medicine, it is also effective that you know that this this medicine is given orally, this medicine is given intradermally, this is given intravenously. So the route of administration is also matters. Microbial load is also matter, that how much microbial load is present in the body of the patient. So according to the microbial load, we have to decide the antibiotic load, right? Patient immune system also matters, that if the patient is able to bear that particular antibiotic or not. So according to these things, we are able to influence the effectiveness of the particular antibiotic, right? Some examples of antibiotics are sulfonamides, which inhibit the folic acid synthesis, quinones, which inhibit the DNA gyrase. It means they inhibit the synthesis of the nucleic acids of the bacteria. Penicillins, which inhibit the cell wall of the bacteria. So without cell wall, the bacteria is not able to infect any other tissue. Other is drug resistance. Drug resistance defined as the resistance which developed from the drugs, right? If you are taking a particular antibiotic for a particular infection, right, and you stops the course of the antibiotics, so after stopping the course of that particular antibiotic, the bacteria acquires the mutations and it develops the resistance to that particular drug. This is known as drug resistance. It means the bacteria is not sensitive to that drug. Now it is resist to that drug, so you have to use another drug. So there are some natural resistant or also acquired resistance. In natural resistance, we have some inherited resistance. In natural resistance, bacteria have inherited resistant of some species, and acquired resistance is the resistance which bacteria acquired by mutation or by gene transfer. For gene transfer, bacteria have the plasmids. Plasmids are the extra chromosomal DNA which is responsible for the mutations and for the gene transfers. So this is about your unit 4, which is antimicrobial drugs. So see students, in the subjects of pathogenic microbiology, you have to learn about the pathogenic microbiology intro. You have unit 1, which is about the infectious diseases, that what are the infections, what are the diseases, how the diseases progress, what are the symptoms, how the symptoms arises, when the symptoms arises, what is the virulence power of the microbes, what are the microbes of medical importance, what is normal flora, how the microbes transmit, how they cause the infections, what are the routes they takes to penetrate the body of the host, right? In unit 4, you learned about the antimicrobial drugs, that what are the antimicrobial drugs, how they develop, who is the father of the antimicrobial surgery, antimicrobial drugs, what are the general characteristics of the antimicrobial drugs, what is the determination level of antimicrobial activity, what are the actions of antimicrobial agents, what are the factors that influence the effectiveness of antibiotics, and what is the antibiotic resistance. So I hope it is clear to you that the pathogenic microbiology is a subject in which you learn that what are the microbes, how the microbes comes inside the host body, how they spread the infections, what are the route they choose to cause the infections, how we are able to treat the infections, how we are able to identify that particular antibiotic is effective against the particular microorganism, and how different routes of the antibiotics which we use to suppress the growth of the antibiotics. I hope it is clear to all of you. So thank you so much for listening the class. I hope the class is beneficial to you, and the concepts which I have shared with you that are clear. If you have any doubt, you are allowed to write your doubts in the chat box, and the unit wise videos are also available on your LMS. To get the more detailed knowledge of any unit, you have to go to your LMS and watch the unit wise videos, right? So thank you so much. If you have any doubt, just write your doubts in the chat box. I will answer your queries. we have two types of bacteria, gram positive bacteria, gram negative bacteria. और on the basis of cell शेप के बेसिस पे अगर मैं बात करूं तो हमारे पास मल्टीपल टाइप्स का बैक्टीरिया है, कुछ कोकाई है, कुछ रॉड्स हैं, कुछ बेसिलाइज है, और आपका कुछ स्पाइरोचीट्स हैं, कुछ माइकोप्लाज्मास है, कुछ एक्टिनोमाइसीट्स है. इट मींस उनका स्ट्रक्चर कैसा है, स्ट्रक्चर के बेसिस पे हम माइक्रोब्स को बांट सकते हैं, राइट. और हम उसके सेल वॉल के बेसिस पे बांट सकते हैं, जेनेटिक मटेरियल के बेसिस पे बांट सकते हैं कि माइक्रोब में कौन सा जेनेटिक मटेरियल है, क्या माइक्रोब में डीएनए है, क्या आरएनए है, क्या दोनों है. और दूसरा कि माइक्रोबियल का मोड ऑफ न्यूट्रिशन क्या है, कि वो न्यूट्रिशन कैसे ऑब्टेन कर रहा है, क्या वो ऑटोट्रॉफ है, अपना खाना खुद बना रहा है, क्या वो फोटोट्रॉफ है, अपना खाना लाइट से बना रहा है, क्या वो हेट्रोट्रॉफ है, अपने फूड के लिए दूसरों पर डिपेंडेंट है. सो इन सारी चीजों को यूज करके आप किसी भी माइक्रोब को क्लासिफाई कर सकते हो. तो यूनिट 2 इज अबाउट द क्लासिफिकेशन ऑफ माइक्रो ऑर्गेनिज्म. What do you mean by chemotherapy? See, therapy means treatment. So when we are using the chemicals to treat the particular disease, it is termed as chemotherapy. When you are taking the support of chemicals to treat the specific disease, it is termed as chemotherapy. The chemicals which we are using to treat the patient. एंड जिसको समझ नहीं आया, कोई डाउट है, इंग्लिश में कोई कॉन्सेप्ट नहीं समझ आया, यू आर अलाउड टू आस्क इन द चैट बॉक्स, आई विल आंसर. की हेल्प से आप किसी चीज को ट्रीट कर रहे हो, दैट इज कीमोथेरेपी. बेसिकली सी स्टूडेंट्स, कीमोथेरेपी हम यूज करते हैं कैंसर पेशेंट्स के लिए. उसमें हम प्योर केमिकल्स का यूज करते हैं कैंसरस सेल्स को किल करने के लिए, राइट. तो कीमोथेरेपी ड्रग्स जो होती हैं, जो प्योर केमिकल्स से डिराइव्ड होती हैं, दे आर यूज्ड टू फाइट अगेंस्ट द कैंसर्स. जेनेटिक के बेस पे आप आइडेंटिफाई कर सकते हो. देखो, बैक्टीरिया तो एक ही टाइप का होगा, बैक्टीरिया में तो डीएनए आरएनए दोनों होगा, बट अगर आप माइक्रोब्स की बात करो, माइक्रोब्स को जेनेटिकली बेसिस पर आइडेंटिफाई किया जा सकता है, क्योंकि जो वायरस होते हैं वो या तो डीएनए कैरी करते हैं या आरएनए. तो अगर हम माइक्रोब्स को क्लासिफाई करें तो माइक्रोब्स को आप जेनेटिक बेसिस पे क्लासिफाई कर सकते हो, क्योंकि कुछ माइक्रोब ऐसे हैं जो डीएनए कैरी करते हैं और कुछ आरएनए, और कुछ माइक्रोब ऐसे हैं जो बोथ कैरी करते हैं. अगर बैक्टीरिया की बात करो, बैक्टीरिया के पास तो दोनों है, तो उसको तो आप डीएनए आरएनए दोनों में कंसीडर करोगे. अगर आप वायरस की भी बात करो तो वायरस आपका डीएनए आरएनए दोनों टाइप का है. कॉगलिस टेस्ट आपका बेसिकली जो है बैक्टीरिया आइडेंटिफिकेशन के लिए यूज किया जाता है. See, not only chemotherapy, not single chemotherapy we are using for the treatment of microorganism. For treating the microorganism infection, we are using antimicrobial chemotherapy, right? Basically, they are the chemicals which are used to cure the infections which are caused by the pathogens. टेस्ट है ना, वो आपका स्टाफाइलोकोकस ऑरियस को स्टाफाइलोकोकस की अदर स्पीशीज से अलग करने के लिए यूज करते हैं हम, टू आइडेंटिफाई, बिकॉज जो स्टाफाइलोकोकस ऑरियस होता है इट स्ट्रांगली catalase positive, so to differentiate between the staph other species from the staph aureus we are using the Coagulase test. Yes, normal flora are the microorganisms which are present inside our body from birth, and they are not harmful to us, they are beneficial to us. Microbial adhere means attachment of microbial cell from the host cell. Microbe का host cell से attach हो जाना is microbial adherence. How many types of microbiology? See, microbiology is a vast branch. We are not able to cover the microbiology in single lab. For example, if you are a lab technician and you are working in the lab, so we have parasites, we have fungus, we have parasites, bacteria and the virus. So is it possible to cover all the microbes in single lab? No, because every microbe have different techniques to identify. So microbiology is divided into bacteriology, parasitology, mycology, phycology and the parasitology. So microbiology is like a tree and the tree have several branches. How to treat about the chemotherapy? ग्राम पॉजिटिव बैक्टीरिया आपके होते हैं स्टेफाइलोकोकस ऑरियस, स्ट्रेप्टोकोकस पायोजीना, स्ट्रेप्टोकोकस निमोनिये, एंड ग्राम नेगेटिव में आपका आता है ई कोलाई, सैल्मोनेला, शिगेल्ला. ग्राम पॉजिटिव मींस वो बैक्टीरिया जिसका जो सेल वॉल है उसमें थिक लेयर है पेप्टिडोग्लाइकन की, राइट, और टिकोइक एसिड प्रेजेंट है. और ग्राम नेगेटिव बैक्टीरिया में पेप्टिडोग्लाइकन की लेयर थिन होती है, अबाउट 2 माइक्रोमीटर होती है, जबकि आपके 2 नैनोमीटर होती है, जबकि आपके ग्राम पॉजिटिव में 40 to 80 नैनोमीटर होती है. और ग्राम नेगेटिव बैक्टीरिया में लाइपोपॉलीसेकेराइड्स प्रेजेंट होते हैं, टिकोइक एसिड का कंटेंट कम होता है एज कंपेयर टू ग्राम पॉजिटिव. ग्राम पॉजिटिव में पेरिप्लाज्मिक स्पेस प्रेजेंट कम होता है, बहुत कम. ग्राम नेगेटिव में पेरिप्लाज्मिक स्पेस ज्यादा प्रेजेंट होता है. तो ग्राम पॉजिटिव और ग्राम नेगेटिव हमने सेल वॉल के बेसिस पे आइडेंटिफाई किया हुआ है. मॉन्टेक्स टेस्ट बेसिकली टीबी के माइकोबैक्टीरियम ट्यूबरक्लोसिस को डिटेक्ट करने के लिए यूज किया जाता है. इसमें हम एक प्रोटीन प्यूरिफाइड डेरिवेटिव इंजेक्ट करते हैं पेशेंट की बॉडी में. अगर पेशेंट कभी एक्सपोज हुआ होगा माइकोबैक्टीरियम ट्यूबरक्लोसिस की प्रोटीन से, तो आपका जो वो प्रोटीन आपने इंजेक्ट किया है उसके अगेंस्ट इम्यून सिस्टम एक्टिवेट हो जाएगा, और उस जगह पे जहां पे आपने प्रोटीन प्यूरिफाइड डेरिवेटिव को इंजेक्ट किया है वहां पे स्वेलिंग देखने को मिलेगी. अगर स्वेलिंग का डायमीटर 5 mm से ज्यादा होगा, इट मींस द पेशेंट इज हैविंग द माइकोबैक्टीरियम ट्यूबरक्लोसिस इन देयर स्ट्रीम, इन देयर ब्लड स्ट्रीम. He is infected with the microorganism. I hope it is clear to everyone. Detailed unit-wise videos are available on the LMS. Just go through your LMS and watch the videos and collect your doubts and ask in the live classes. So, thank you so much for joining the class. Now you are allowed to leave.